Hello, everyone, and welcome to this new edition of the Impactive webinar series. It's a pleasure for me to be here today with all of you in another one of our fantastic webinars. Like you know, Impactive is a project funded by the European Commission through the Horizon Europe program, and we basically mash molecules together. We work in mechanochemistry as a solution towards greener pharmaceuticals in industry and worldwide in general. So what we're doing in this series of webinars, among other things, is also establishing connections and building bridges with other projects in Europe working with the same goal of making pharmaceuticals greener. And today it's a great pleasure and a great honor for us to have Christian Stevens joining us. He's the coordinator of a sister project, TransPharm, which is also looking into greener pharmaceuticals. And he's also currently senior full professor at the Department of Green Chemistry and Technology in Ghent University in Belgium. And his research focuses in things like microreactors, synthetic chemistry, synthetic heterocycles, and in particular, the developing pharmaceuticals and agrochemicals for cool applications and green applications using renewable resources in the lab and hopefully soon in industry as well. So TransPharm is also funded by Horizon Europe, like Impactive, and their aim is to move forward a greener pharmaceutical industry, and specifically the project works, to improve Europe's capacity in the sustainable production of APIs, which is an acronym. Scientists love acronyms. An acronym for active pharmaceuticals ingredients. In particular, those that may have some environmental or supply concerns, which, as you know, especially after the COVID pandemic, supply of pharmaceuticals is a very key and strategic point for the European Union, because we need to make sure that we can produce pharmaceuticals without relying on other. Just before we start, and I give the floor to Christian, I want to remind everybody that this is streamed live on YouTube. So if you have any questions, we'll have a little Q&A at the end with Christian. Please leave them in the chat on YouTube. There's a space there for you to leave questions. I'll be checking at the end to pass them on to Christian. And if you don't already, make sure that you follow Impactive on our webpage and social media. We're on mechanochemistry.eu, and then I think X, LinkedIn, the usual social media. We also have a fantastic newsletter every couple of months. Again, we try to make this super interactive. Of course, it's going to stay on YouTube forever, but if you want to become a part of the conversation, maybe drop a message in the chat so we can bring it up later on. And with that, and without further ado, I just want to leave the floor to Christian. Christian, very happy to have you here. I'm really looking forward to your presentation. And yeah, of course, the floor is yours.
Thank you very much, Mr. Chairman, for this nice introduction and for explaining already a little bit what the Europe Horizon project is doing. So indeed, we are the TransPharm project, which is a sister project of Impactive. And in the following 20-25 minutes, I will try to explain what we are doing in this European project. So we are indeed trying to transform the pharmaceutical sector in a sustainable way. And our ambition and vision is to really improve the EU readiness for sustainable production. And we're concentrating on small molecules. So we're not dealing with proteins, with nucleotides, and so on. So not with biologicals, but we are focusing on small molecules, mostly heterocyclic compounds that are used as a drug in the pharmaceutical industry. So the basis of our project was the benign by design principles. So we really want to design from the start of the development of APIs to really look at the design and to see if we can build in some features that are better for the environment or throughout the complete lifetime of the API. So we look at greener pharmaceutical production, involving the choice of solvents using continuous flow chemistry. We look certainly at producing compounds that have a better biodegradability. I will come back to that later on because that's a really important aspect of our project. And we are working also on the assessment of the sustainability. If we make changes, can we also prove that indeed it is better for the environment or for the economy, the bioeconomy, and so on. So we're doing quite a lot of LCA studies as well. And of course, the idea is then to transfer the knowledge to our stakeholders so that they can use it in practice. So the way we set it up is that we formed a consortium with complementary technical capabilities. So we have groups that are really expert in design and synthesis, and my lab is working, as was explained, mainly in organic synthesis of the molecules, but we also look on the environmental fate, LCA as said, and the impact on stakeholders. And of course, as in every EU project, also communication and the management is quite an important aspect. So that's an overview of the partners. I will not go through the complete list, but as you see, they are spread all over Europe. We have, of course, Belgium, we are coordinating, but also Finland, Norway, the Netherlands, Germany, UK, Ireland. So we really have a very complementary group working together on this important topic. So our major concern is the fact that a lot of the production of pharmaceuticals nowadays have been shifted to lower income countries. And that leads to a number of problems. Of course, the instability of production, unreliability of supply, and above all, also the lack of global environmental protection if we want to bring back this production to the EU. One of the main problems is if we just copy the kind of production systems from these lower income countries, that we end up with a quite heavy impact on the environment. And it also, in our case, just by the regular use of APIs, we see that the APIs are very much present in the environment, and mainly because of the fact that a number of APIs are not broken down completely in the wastewater treatment systems. They end up in the environment, and specifically for antibiotics, it leads to multi-resistance of bacteria, and that's of course a major problem in the EU. And studies predict that if we don't do anything about that, it will be a major problem 50 years from now with a lot of multi-resistant bacteria. So that's really something we have to act upon. And that's why we're trying to design new molecules that have a better biodegradability, but of course, keeping the activity of the compounds. And this idea is completely in line with the Green Deal that is promoted by the European Union, and also the intention to bring back pharmaceutical production to Europe so that we have a security of supply, which is also a major problem for a number of very normal kind of APIs. So the project objectives are in two sentences: the fact that we want to develop environmentally friendly APIs, and all over the lifetime of the API through production and use, we want to reduce the environmental impact. And that is in line with the global sustainability goals of the UN. And you see on the bottom of the slide 8 of these rules that are taken care of, or that are associated also with activities in our TransPharm project. So how is the project structured? We have 7 work packages. First, on the use of data that are available in the literature. We don't have to find out things that are already there, so we want to maximize the data use. Then we want to use that in the development of better biodegradable compounds. using greener pharmaceutical production tools, and this will be covered or will be studied to see that it really makes a difference in the integrated sustainability assessment. And then also very important aspect in our project is the fact that we want to check if the society is ready to adopt the use of greener pharmaceuticals. That's also a very important aspect, that we do not develop technological tools that at the end of the road nobody wants to apply because people are not interested to shift to the use of more green pharmaceuticals. So there must be an interaction with all stakeholders to make sure that at the end of the day, people want to adopt and want to use this new materials. And if you see on the left of the slide, the work package 1 with the data use is overarching the 3 other work packages, and there is a very strong interaction between all the work packages, and that leads to work package 5, where we also make a case study for one of the APIs. So again, I'm not going into details here, but it shows that in every work package, we have a very close collaboration between academic partners as well as companies in order to promote the production of the greener pharmaceuticals. On top of that, we have also an advisory board, which is composed of a number of big pharmaceutical companies, such as Boehringer Ingelheim, Takeda, and Johnson & Johnson, but also other institutes such as Sustainable Healthcare Coalition, FPA, and Healthcare Without a Harm. And it's the idea that these people really keep us on track and give us information on what is really needed in the field, so that we really focus on the important aspects of the industry, of the pharmaceutical industry. I will now share some more detailed information on the different work packages to give you a flavor of what we are really doing in the different work packages. Of course, I cannot go into too much detail, but at least it will give you an idea. So work package 1 is focused on the data use coming from literature. And here we are developing systems based on machine learning and quantum chemistry to extract new data from the literature. So in the past, these models that were used for structure-activity relationships were based on the features of the molecule. So you feed in the structure of a molecule, and then a number of physical chemical parameters were calculated for every compound. That could be molecular weight, it could be the amount of hydrogen donors, the amount of hydrogen acceptors, the polar surface area. So a few hundreds of different features were calculated for every compound, and from that, a regression algorithm was produced to predict the molecular property of some unknown compounds that were later fed into the system. But here in these older systems, the mathematical description of the molecule is the same in every situation. And that we now changed to a new system, a computer learned representation. So it doesn't work with features, but the system learns itself to have a better molecular representation. So the mathematical description of the molecule is optimized during the learning process, and that gives a better prediction, but also the fact that the prediction has a better speed, a better accuracy, and also interpretability. So that has been published in a number of publications to use that open access information. We did actually the same for reaction routes. So for reaction schemes, where there was a system to predict the experimental conditions of a flow process. So again, from the literature data, a system was developed to be able to handle a reaction so that the computer system realizes which functional groups are going to react in a certain reaction that goes through an atom mapping, and the system recognizes which functions can react. That leads to a reaction classification, and finally, from that, from the literature, we get suggestions on the conditions that we should use if we want to make a new compound in flow. Of course, this is also related what is present in the literature. So if a certain reaction is always done, for example, in dichloromethane in the literature, the chances that that comes out in the prediction is also bigger. So there is also some chemical knowledge needed to interpret also the reactions that were given through the system. But this is a completely new way of trying to speed up the development of organic reactions. In work package 2, we are working on developing more biodegradable compounds. And here, as one example, I can give the antibiotic ciprofloxacin, which you see on the screen, and you see that on the nitrogen you have a cyclopropyl substituent, and that hinders the biodegradation. And we have prepared a number of derivatives which are similar to the one presented on the right, the SEP-HEMI, and here you see that the substituent is a tetrahydrofuran compound, which makes a hemiaminal. And as you know, hemiaminals are very labile in acidic media. So once these kind of compounds come into the wastewater treatment system, it hydrolyzes very quickly and leads to an inactive compound, and that leads to a better action, so that no resistant bacteria will be formed against this antibiotic. So we made a complete family of these compounds. First, they were predicted by an in silico method, which was developed by the University of Leuphana in Germany. And we worked on the organic synthesis, and that was then treated and studied for the biodegradability. Also, a lot of work has been done on hardware to measure the biodegradability more automatically. So University of Helsinki has been working on a pilot scale system to do automatic oxygen sampling and automated sampling. So we need less compound to measure the biodegradability, and we can do it in an automated way, so that you can also do more frequent measurements, and that can lead to a better understanding of the kinetics of the biodegradation. Work package 3 is then working on renewable building blocks. So it's also very important to try to use renewable building block instead of fossil building blocks. And here we are doing a lot of work on LGO. It's levoglucosenone. It's a derivative that also leads to the biodegradable solvent styrene. And here we are doing quite a lot of fundamental work. As you see, we have a sequence of biofilic oxidation and protection, and then we can study all kinds of different Michael additions to have a broader set of building blocks which are derived from a natural resource. In this case, it comes from wood chips. Also, our partner in Latvia is working on electrochemistry as a tool to do the chemistry in a more sustainable way. Here they're working from furan type derivatives to do a ring opening reaction to give an acetal, and that is further worked on to the corresponding acid, which is then derivatized with a chiral auxiliary, and that leads to the possibility to do enantioselective reactions on these natural building blocks onto the way of some very specific gabapentinoids that are useful in the pharmaceutical industry. Work package 4 is the work package on the life cycle assessment. So here we built on a holistic framework for assessment. And one of the major problems is the fact that there are a lot of data gaps in the LCI data. So these have to be predicted first in order to be able to have a good LCA method to evaluate the APIs. But we are also working on methodological innovation. For example, the spatial dimension, it means where does the API gets into the environment and what will be the influence on the place where it gets into the environment. Situation in Italy will be different from Germany, from the Netherlands, because of temperature, because of the hours of sunlight. Does it get in a river? Does it get in a lake or in the sea? So the spatial aspects will be very important also on the biodegradation of the compounds. We are looking also on the effects of combined impact. That means what is the effect if we have two antibiotics coming into the environment, and also work has been done on expanding the characterization factors for ecotoxicity for more than 250 APIs. So that has been done, and that work is now being used to look at a case study in a hospital in Switzerland where the wastewater contains a number of excreted antibiotics. We look at the wastewater treatment, what remains in the wastewater after treatment, and then we try to evaluate what is the effect on the environment of the combined antibiotics. So we look at freshwater ecotoxicity impact. And at work package 5, we look at how we can deal with all the stakeholders. From a number of interviews with experts, API producers, formulators, supply chain specialists, we try to see how easy it would be to uptake a greener pharmaceutical in the supply chain. And an important review has been published on the keystones in the implementation of greener pharmaceuticals that you can find back in the journal Sustainable Chemistry and Pharmacy. We had also meetings with the European Medicine Agency to discuss the hurdles that we hear in this whole process, to see how EMA can act on this and how this can be brought to the pharmaceutical industry. In the same work package, we also work on continuous flow chemistry to try to improve the batch chemistry, again, without going in details, but this is work on ciprofloxacin derivatives. We developed a new method to do the N-alkylation, and we have proven that we can improve the volumetric productivity with a factor of 230. So indeed, we also know from the LCA work that switching from batch to continuous flow really improves or reduces the environmental impact on the environment. And then finally, in work package 6, we are developing a lot of educational training material that will come online very soon. It's called SPLASH, Sustainable Pharmaceuticals Learning and Advanced Science Hub. And here we brought together a lot of information on how pharmaceuticals are being produced, what are the technologies that are being used, how important is the influence on patients and so on. And as you see, We developed this training material for different stakeholders. We developed it for healthcare professionals, for R&D and academia, but also for members of the broad public who do not know a lot of basic or do not have a lot of basic knowledge on the pharmaceutical industry, and that will come online very soon. So to conclude, our TransPharm project has a duration of 48 months with a budget of about 8 billion euro. And as said by the chairman, there is indeed a collaboration also with the other projects in the Horizon Europe called CEST, Eternal, IMPACTIVE, and Environment. And we're also working together now with the follow-up project, Farm Eco, which is also a European project with a lot of industrial partners. And we try to interact so that the two projects can benefit from the results of each other, so that we're not duplicating the work and that we're helping each other to move this important challenge further on. So by this, I want to thank you for your attention, and I'm more than happy to answer your questions on this topic. Thank you very much.
Thank you very much, Christian. It was a lovely presentation, and I've been very pleasantly surprised. We're just commenting like offline how amazing it is that you're also developing educational resources. That's something we just launched at Impactive. We have a new educational platform about mechanochemistry. So maybe we can take the discussion offline. Maybe we can have a chat about the potential collaborations in education, because I think it's a very interesting topic to educate not only the wider public, but specifically policymakers on the importance of supply chains, sustainable pharmaceuticals, green pharmaceuticals. So maybe we can have a separate chat about that. And honestly, I love your presentation. I made some notes on the way. I loved even like the deep chemistry slides. You had some very nice multi-gram examples. I love the example of the new antibiotic that breaks down in water with the new cycle. Instead of the cyclopropane, it has a little oxygen, and then that makes it break more easily. And I also love like seeing, relatively new techniques like flow chemistry applied in the manufacture of pharmaceuticals. I was especially curious about one of the things, because I think it was World Package 2 or 3, you mentioned new machinery and new equipment to test the biodegradability of components and chemicals. And it reminded me of, when you go to IKEA, the machines they have to test the chairs and the mattresses like several times to see if they're resistant. So I think that's a parallel with what you're doing. But could you give a little bit more of an in-depth explanation of what you're looking for in those machines and how they work, and how do you analyze the fragments generated?
So what we really want to develop is it's similar to the closed bottle test, where you can test for the biodegradability of compounds. So these are tests where you measure the biodegradation for 28 days. And of course, we want to speed up this reaction, and we want to automate it. So the problem is with the closed bottle test, you normally need quite a lot of compound to do the testing. And so for every compound, you have to produce a certain amount of compound. By trying to miniaturize the system, we need less compound to be produced. And of course, that goes hand in hand with continuous flow chemistry, where we can very quickly make the compound, and that can be tested. And so we can measure the oxygen uptake during the biodegradation, and that is, of course, the measure to know how fast this biodegradation takes place under an oxygen atmosphere. Of course, there are some technical problems, because the consumption of the oxygen is also a lot smaller because you have so little compound. So the measurement should be very, very accurate to have a good view on the biodegradability of the compound. So there are a number of technical issues, but We solved most of them, and now we are trying to make a better machine so we can test like 8 compounds at the same time.
And how do you this may be just my past chemist wondering, how do you test for fragments like after the solution has been exposed to oxygen during like the 28 days? What do you look for? Like, do you look into the different fragments that have been generated? Do you look
Yeah,
into whether they're
we
dangerous or sustainable?
Yeah, well, the most important aspect, of course, is that you want to be sure if you biodegrade compound that you don't produce a more toxic compound, which is possible. I mean, you never know if you degrade the compound that it gets worse. So
That's a
you need
classic.
yeah, that's a classic. So you need to make sure that you indeed lower the toxicity and that there is not a problem. So we look in a further stage also to the degradation products. And of course, in the end, you want a complete mineralization of your compound. For example, what we see in the case of the ciprofloxacin, we don't get a complete mineralization, but we know that degradation products are not more toxic than the parent compound.
I mean, that's really interesting. I'm just like amazed by how can you look at the different fragments, because obviously you start from one API and you must end up with a relatively messy mixture of products, and that's always hard to deal with. But it's really interesting. I was also very keen on the first slides on, I think it was what package one, where you had all the in like improved and enhanced machine learning models to sort of use different predictors and different data points from molecules to design better molecules from scratch. And I thought that was quite interesting. How exactly do these models differ from the models available already,
Yeah,
and what makes them better? And in the sense of like, do they give more points to sustainability, or to maybe sustainable by design structures that are easier to break, that like the antibiotic that you showed?
yeah, it's a good question, but of course, I'm not the expert in artificial intelligence. I'm more a synthetic chemist. But what we see is that actually the new system uses a very old technique where it was proven that you don't need the 3D structure of the molecule to have a good prediction, that a 2D structure gave the same quality of prediction. So that reduced enormously the time that we needed to calculate all the compounds. So the only difference is that in between the work that has been done by the algorithm, this becomes really a black box. You don't know what's happening there, but we realize that the predictions are better and that it can be done on much less time, and that's of course important. And now we can feed in new molecules that are not synthesized before, and it predicts if this will be better biodegradable or not. So we are now working on systems that will also deal with biodegradability of the compounds. One challenge there is that you need to train the molecule, that you need to train the model on data that you have from these closed bottle tests. And the number of APIs where you have the full data set are rather limited. And so we are working on that in order to improve and to see if the predictions are really in line with what we find if we do it in the miniaturized system.
I think that that sort of ties perfectly with the next question that I had, which is related to data points you were just mentioning. Because I've seen in some of the slides that you're making most of the algorithms and programs open access through platforms like
Yeah,
GitHub.
yeah,
But what about the data? Is that available somewhere? I'm thinking Zenodo, or maybe things like IOChemBD, like public databases with the
yeah.
results of simulations that could be potentially access in the future to be analyzed or mined by other
Yeah.
consortia to make the data even more available?
Yeah, that's a very good question, and it's a challenge that the five projects are struggling with because when we develop LCA methodology, you want to have like a validation. And of course, because a lot of these data on APIs are confidential, they are not completely available, or even the production data are not available or are not shared by pharmaceutical companies, which is normal. So that was one of the points that we have been discussing with EMA, with the European Medicines Agency, that they should try to help on a European level to get for at least one or a couple APIs all the data that are needed to really validate these LCA models, because every project is working on innovation in LCA, but it's very difficult to compare the different LCA models. And if we would have a set of one API where we have all the data, we would be able to compare the different LCA models and see which one is working better.
Uh-huh.
But at the moment, this is not available, and we're also discussing with a number of big pharmaceutical companies to see which data we could use to do a good validation of the system.
I think that's an excellent point because I remember a while ago attending a conference from like car manufacturers, and they had the same issues with like LCA points because some people go with mileage, some people go with emissions, and then obviously depending what you measure, you can give fake green measurements,
Yeah,
sort of whatever you want, you can make it green. And I just came back from a conference in on hydrogen last week, and the discussion was the same exact discussion on like finding an agreement on what's green, what's
yeah.
not, and what manufacturing. So I think that's an issue we're gonna encounter, especially in Europe with so much regulation. So I think it's very important that you deal with policymakers from the project and from the experts directly. So what strategies are you following? I saw you've got like a work package dedicated exclusively to that, but what strategies are you following to engage with the European Medicines Agency, the AMA and EMA and Brussels? Like, do you talk to them often? Do you organize sessions or breakfasts or meetings with them very often?
No, not very often, because it's not that easy to arrange a meeting with different sections of the EMA. But we have to be also aware that the pharmaceutical sector is doing a lot of work in order to try to evaluate in the same way. So also from the sector itself, there's a lot of efforts to try to come to a system that everybody can use and where you can really see what's going on. So I think many people think that maybe the pharmaceutical industry is not really taking care of that, but that's not true. They are really doing a big effort to make a greener production of their pharmaceuticals, but of course, and also towards standardizing LCA methods. So there are working groups, and they are working, and we had some meetings with EMA, but of course, it's a slow process. It's all about safety, of course. If you change something, it has an effect on safety measures, and of course, they are very, very careful to change things because it might
Yeah.
give an effect on patients and so on. So it's a delicate a delicate topic to change things and the way they are working at EMA.
Yeah, I think that's probably the main hurdle, to apply the
Yeah,
work of TransPharm and other projects is to how to find a common ground for discussion with the EMA, with the pharmaceutical sector. I think we've covered that, but is there anything you want to add on the hurdles that you've experienced so far
yeah.
to sort of
Well,
translate the knowledge of TransPharm to
yeah.
the big pharma?
One important point that we really gave to EMA that was, to us, it's one of the big hurdles, is the fact that if we find a method, it can be in the production or a new derivative that is better for the biodegradation. But a good example is, for example, if we find that a certain batch reaction can be done in flow and that improves the impact on the environment, the problem is that the companies are not very keen to change their master file, because once the master file is submitted to EMA and FDA and is approved, yeah, it takes a lot of time, a lot of effort, a lot of money to do the changes. And so, yeah, pharmaceutical industry is not keen to do all the work just to implement a greener method because it just takes too much money. And so that's really a point that we brought to the attention of EMA, that if there are minor changes to the production process, but it has an impact on the environment and on the sustainability, the administrative burden to do the changes should be smaller, so that the pharmaceutical industry is keen to do the effort to change the master file and to produce the compound in a more sustainable way.
And it's something that is proven to work. I mean, we saw it during the COVID pandemic that sometimes you can go through the sort of hurdles quicker if there's
Yeah,
need or if there's
yeah,
pressure. It's funny because I think, again, I'm thinking of the collaboration between the sister projects, and I think this is another point where we can have a very interesting discussion with the researchers in Impactive, because obviously for us it's also a big hurdle to transform the pharmaceutical industry into new methodology based on continuous flow methods like extrusion, or maybe introducing mechanochemistry instead of solution chemistry, which, like you said, may introduce some new properties to the molecules that you don't know based on crystallization, based on different aspects of the process. So it's important to control that, but at the same time, it's important to know when to do the transition to avoid further delays. I wanted to sort of move away from hurdles and close with a little bit of a more positive spirit and a more positive question. Is there any cool results, any nice success stories, any great achievements from TransPharm that you want to highlight to close this fantastic webinar and discussion that we had?
yeah. I think the work that we have been doing in this project on this antibiotic is really a nice example that it is possible to design molecules that are better biodegradable and that have the same activity. Of course, we have examples that have a lower activity, and then the discussion changes, of course, because if the activity is a little bit lower, you need to apply more compound to the patient, and so you have to produce more compound, and so you lose because you need to use more resources. So it's a very delicate balance. You really have to design for better biodegradability, but for the same or better activity. And in the case of this ciprofloxacin derivatives, we have been able now to produce compounds that have these features, and I think that's a very, very nice result. And on top of that, also the work that has been done on the closed bottle test to have an automated system that will also advance the study of the biodegradability quite a lot. So there are a number of important realizations. Also, the fact that we can now account for the spatial distribution of API in the environment is important, because there is quite some difference if a compound gets in a lake in Italy and it stays there 10 days in the lake before it degrades, or it comes in a river in Germany and it stays there for 50 days because the weather is different, there is less sun, and so on and so on. We really have to look to regions to account for the environmental burden. So that's also something that came out of the study, and which I think is also a very nice realization, next to the fact that, but that's then more technical, that we developed some very interesting synthetic methodology, for example, for oxidation of alcohols and so on. There are many examples of synthetic work, but that should lead us in a very technical discussion.
Yeah, I had a couple of questions about skeletal editing because I've just been reading a lot about that, and I had like, it could be interesting to look into the opportunities for this type of reactions as well. But I think I was getting a message right now, like offline, saying like this is a little bit too long for the YouTube algorithm, so maybe it's the perfect time to stop. But honestly, I wouldn't want to close without like, first of all, thanking you again for your time and a wonderful presentation, but maybe most importantly, I want to highlight the room for collaboration between TransPharm and IMPACTIVE in so many different areas. We face the same hurdles in translating our chemistry to the pharmaceutical scene. We probably have a lot in common in the educational arena. We're both trying to create new materials to reach out to the wider public and policymakers. So there's a lot of room for collaboration there. And I think also we all have the same dream, which is developing greener pharmaceuticals and communicating about that, because that's what we're doing with this webinar, and that's what you highlighted. That's one of your main work packages, being able to translate the results into policy briefs, into information for pharmaceutical companies. So I mean, that's something that we do a lot in our company in AGATHub, and of course in the project in IMPACTIVE. So it'd be fantastic if we could potentially sit down and talk offline. I remember when we first met the projects I think it was in Nijmegen in the Netherlands. We had a little meeting between the projects, and we were all a little bit lost because we had just started a few months prior. And now seeing all the results, seeing all of the developments, and seeing all the advances, it really looks like money well spent from the European Commission. So I'm very happy to have had the discussion with you. Thanks again for joining us.
With pleasure, and I hope we can continue to collaborate between the projects, because I guess that also in the history of the European Commission, it's for them also one of the first times that they see such a close collaboration between different projects aiming for the same goal, and that's exactly what is needed in Europe to advance the field, I guess.
I mean, I think it's been a fantastic discussion, and we've really enjoyed having you here. We go very often to Brussels. It'd be fantastic if we could, take a further train to Ghent and maybe visit your labs, have a more detailed conversation about this. But again, we could do that offline. I just wanted to thank you again for joining us, and just a tiny acknowledgement for everybody, watching us. Thanks for joining. Thanks for joining live. Thanks if you join later on YouTube. We are part of IMPACTIVE, which is a project funded by Horizon Europe. I know I said at the beginning, but maybe you skipped that part. You can find all of the information about the project in www.mechanochemistry.eu. And you can also subscribe to our social media and newsletters if you're interested in more news about green pharma and mechanochemistry. You'll hear about us, but then also about our sister projects like TransPharm. Please don't hesitate to contact us if you have any questions. Thanks for joining us again. And yeah, looking forward to seeing you on the next webinar, which, if I'm not mistaken, is next week. So for all of you, watching us live, join us next Wednesday. We have another sister project explaining the technologies here on YouTube. So thank you so much again for joining. And yeah, see you on social media and see you soon.